Welcome to my YouTube channel. Today we'll be discussing phases of the bacterial growth curve. So, what is the bacterial growth curve, and what are the phases, and what that does each particular phase of the growth curve represent? So, the bacterial growth curve graphically it represents the number of living organisms or living cells in a bacterial population over time. So this one can be used, like we can say it's like a predictable pattern of growth when bacteria are grown in culture. So as we all know, bacteria are prokaryotic organisms that mostly replicate by the asexual process of binary fission. So these microbes reproduce rapidly at an exponential rate under favorable conditions, resulting in the predictable pattern represented by the growth curve. So bacterial growth curve, it is consists of four phases. We have the lag phase, the exponential phase, the stationary phase, and the death phase. So, what happens during the phases of the bacterial growth curve? So, in nature, bacteria do not experience a perfect environmental conditions for growth. So, as such, these species populate an environmental change over time. But now, in a laboratory, optimal conditions can be met by growing bacteria in a closed culture environment. So it is under these conditions that the curve pattern of growth of bacterial growth can be observed. So the bacterial growth curve represents the number of cells in a bacterial population over a period of time. So let's move on to the phases of the bacterial growth. So the first phase we are going to have the lag phase. So the lag phase is the initial phase and it is characterized by cellular activity but not growth. So a small group of cells are placed in a nutrient-rich medium that is going to allow them to synthesize proteins and other molecules necessary for replication. So these cells are going to increase in size, but no cell division is going to occur in this phase. Then now we move to the exponential phase. So after the lag phase, bacterial cells are going to enter the exponential or log phase. So this is the time when now the cells are dividing by binary fission and doubling in numbers after each generation time. So during this phase, metabolic activity is usually very high because the DNA, RNA, cell wall components, and other substances which are necessary for growth are generated for division. So during this phase, uh, antibiotics and disinfectants are most effective as these substances typically target bacterial cell walls or the protein synthesis process of DNA transcription and RNA translation. Then we move on to the stationary phase. So eventually, the population growth experienced in the log phase, it is going to begin to decline, and the decline of this phase is because now the available nutrients become depleted, and we are going to have waste products which start accumulating. So this accumulation of waste products is going to bring out a result of the decline. So we are going to have the number of dividing cells are going to equal the number of cells which are dying. So as a result, we are going to have there is no overall population growth during this their phase. So under less favorable conditions, we are going to have competition for nutrients and the cells become less metabolically active. So we are going to have spore forming bacteria produce endospores in this phase, and pathogenic bacteria begin to generate substances, that is the virulence factors, which are going to help them to survive harsh conditions, and consequently we are going to have a disease being caused as a result. Then we move on to the death phase. So during this phase, as nutrients become less available and waste products increase, the number of dying cells continue to rise. So in the death phase, the number of living cells decrease exponentially and population growth experience a sharp decline. So during this phase, you are going to have cells, dying cells are going to lyse or break open. We're going to have their contents being spit into the environment, so making these nutrients available to other bacteria. So this is going to help spore forming or spore producing bacteria to survive long enough for spore production. So spores can survive the harsh conditions of the death phase and become growing bacteria when placed in an environment that supports life. So now the question is, do bacteria really need oxygen to grow? So bacteria, like all living organisms, they are going to require an environment that is suitable for growth. So this environment must meet several different factors that support bacterial growth. So which factors are these? So we're going to have factors like oxygen, pH, temperature, and light requirements. So each of these factors may be different for different bacteria, and it is going to limit the type of microbes that are going to grow in a particular environment. So bacteria, this one now calls for bacteria being classified or categorized based on their oxygen requirement or tolerance levels. So, bacteria that cannot survive without oxygen are known as obligate aerobes. So, these microbes are dependent upon oxygen as they convert oxygen to energy during cellular respiration. So, unlike bacteria that require oxygen, other bacteria cannot live in its presence. So these microbes are called obligate anaerobes, and their metabolic processes for energy production are stopped in the presence of oxygen. So we have other bacteria which are facultative. So facultative anaerobes can grow with or without oxygen. So in the absence of oxygen, they utilize either fermentation or anaerobic respiration for energy production. So aerotolerant anaerobes are going to utilize anaerobic respiration but are not armed in the presence of oxygen. So microaerophilic bacteria require oxygen but only grow where oxygen concentration levels are low. So Campylobacter jejuni, for example, is an example of a microaerophilic bacterium, and it grows in the digestive tract of animals, and it is a major cause of foodborne illness in humans. So let's move on to bacteria growth and pH. So how does pH affect bacteria growth? So another important factor for bacterial growth is pH. So acidic environments have pH values that are less than 7. So neutral environments have values at or near 7. And basic environments have pH greater than 7. So bacteria are acidophiles. Bacteria that are acidophiles thrive in areas where the pH is less than 5, with an optimal growth value close to